So if you have any doubt in the light microscope or light microscopy, you can tell me, I can repeat that part for you. Okay. So this is the dark field microscope. Okay. So we can see one image which makes the difference clear. So as you can see in this image, the image on left hand side is of light field microscope, the normal microscope which we use in our labs normally. Okay, this is how the organisms or the objects or the samples will appear. So if you observe, the background is white colored or colorless. It is bright in nature. Okay. As compared to that, if you observe the picture on the right hand side, the background is completely dark. Okay, and the samples or objects are appearing illuminated because of the special set of condenser that this microscope has. You observe something like this in negative staining, okay, when you are going to perform it in the lab, but the cells will not be illuminated. Okay, those will just appear colorless against the black background. Okay, so you will be able to see colorless rods on the surface of a dark black background. Can anyone tell me why nigrosin black will not stain the bacterial cells? Can anyone explain me the reason? If anyone knows the reason, just unmute yourself and answer.
Because we have not heat fixed it, so the bacterial components wouldn't be broken down to take up the stain.
No, that is not the reason. Who was that?
Ma'am, Ayushi.
Okay, Ayushi, that is not the reason. Anyone else?
Ma'am.
Rituja, yes.
Ma'am.
Bolo.
Ma'am, acidic stain will be negatively charged and therefore it will not penetrate the cell because of negative charge on the surface of the bacteria.
Yes, that is correct. So what will penetrate the surface of the bacteria? What kind of stain will penetrate?
Basic dyes.
Basic dyes. For example?
Methylene blue.
Methylene blue, okay.
Crystal violet.
Crystal violet, yes. Methylene blue is a dye, but we don't use it commonly for staining procedures. Crystal violet, yes, most commonly used example. Yes, that is the reason nigrosin black or mordant black or malachite green are certain dyes which are negatively charged, or these are acidic dyes basically, and bacterial cell surface is acidic or negatively charged in nature because of the exposed amino acid group on the cell wall of the organism. Okay, and because of those exposed group, it imparts negative charge to the cell surface of the bacteria, and that is why there will be no bonding if you are applying negative dye on the surface. There will be bonding between negative and positive. So on negative cell surface of bacteria, if you apply positive dye such as crystal violet, there will be a bond formed between the dye molecules and the molecules exposed on the bacterial cell surface, and that is why bacterial cell surface gets stained. That is the principle of any staining. Okay, and in negative staining you use inverse principle. So exactly same reason. For exactly same reason you are using acidic dye, because you do not want to stain the bacterial cells, and why you don't heat fix the organism in negative staining, because you want to observe the morphology. Okay, when you heat fix the organism, the cell undergoes shrinkage. Whenever you are going to perform these experiments in the lab, that time you will observe same organism, Bacillus subtilis, rod-shaped organism. When you observe the same organism by gram staining on one slide and by negative staining on another slide, the negative staining slide will give you larger cells, will show you larger cells as compared to the gram staining, because in gram staining you perform heat fixing. Because of that, cell undergoes shrinkage, and in negative staining, because you want to observe the original morphology of the bacterial cells, and the procedure is such a way that you want to make a smear on the slide, right? So you want to put that bacterial culture in a liquid state, okay, and mix it with the dye, and with the help of other slide make a smear. If it is already dried by heat fixing, you won't be able to make a smear of that on the entire slide. To avoid that, we avoid heat fixing. We prefer air drying. Okay, so once smear is prepared, we simply air dry that slide and then observe under the microscope, and that helps us to observe the original morphology of that bacteria, which is not possible with other staining techniques in which we perform heat fixing. So have you learned about acid fast staining yet? Or cell wall staining or capsule staining? Yes, Kritika. Yes or no? Sorry, what was that? Acid fast सीखा है कि नहीं? Okay. I assume no. So let's go ahead with phase contrast microscopy. Yeah. Phase contrast. So sometimes what happens, as we have discussed so far, so microscopy and staining basically go hand in hand, and I assume that you have learned staining in theory as well as in practicals. Okay? Have you learned staining in practicals yet? Yes or no? Because I was explaining everything, assuming that you have learned these things in practicals. Okay, answer something.
Ma'am, we have learned it in theory.
Not in the practicals. So you have not seen pictures or of the slides of the how it looks after staining, of the gram staining or
Of acidic and gram staining we have seen.
okay, pictures you have seen. Okay. So,
yes.
Okay, fine. Okay. So in gram staining, the primary staining used is crystal violet, right? So the procedure goes like this: first to heat fix the culture on the slide, and then you apply your primary stain, and you allow primary stain to react with the slide, with the organisms for some time. Okay. But the thing is, if you apply more and more amount of crystal violet, it turns toxic for the cell. So any dye, any staining agent that we actually use for staining the organism is actually toxic at higher concentration to the organism. So crystal violet is one of the agents that is used to inhibit the growth of gram-negative organisms. It is antibacterial at higher concentration. Okay. So sometimes it might happen that because of the staining procedures, you will end up killing the organism. Okay. So staining procedures, staining techniques might not be suitable in few cases for few organisms. Okay. So in such case, there is a special type of microscopy which is known as phase contrast microscopy. Okay. Phase contrast microscope in appearance is looks similar to your normal microscope. Okay. But again, just like dark field microscope, it has a special set of condenser and phase contrast lenses. So what is the principle? The basic principle is those set of lenses and condenser, okay, they divert the light rays as compared to the refractive indices of the sample. Okay, so contrast will be created at every level. Contrast will be created between the background, between the slide and the sample above that, between the sample and the cover slip above that. Not just that, within the sample, whichever object, whichever part that sample has, every part will be visible differently because those lenses will be creating contrast between the sample and different parts within the sample. So each and every part will be diffracting the lenses in different directions because of the special set of condenser and lenses that we have in phase contrast microscope, and that is why each of the parts of that sample or object will be visible differently. Okay. So okay, we have a yes. If you can see this picture of amoeba. Okay, so So small granular structures within the cells are also differentiable. Okay, this is not possible by any staining procedure or any light field or any dark field microscope, because they diffract light on a same plane. Okay, but phase contrast microscope will help to diffract the light on each and every plane that sample has. Okay, that is how each object inside, each part of the object will be seen differently in case of phase contrast microscope. And this technique is also useful for organisms which cannot sustain the staining procedure. Okay, whose morphology will change because of the staining.